Okay, so good morning, everybody. And today I'm going to talk about development different biosensors for real-time monitoring of the toxicants in the air and the water. So I will start as Michael said, I'm from post-harvest in Food Science Institute in the post-harvest department. I'm here in the Volcani already almost 5 years. Okay, so it's not working. Okay, so I will start from a problem, a big problem, worldwide problem, pollution, air and water pollution. And the problem will only increase during the years because many different new chemicals are developing and finally coming way outside from the industry to environment and then to us. So we need it's affecting us. Actually, every year more people die from unsafe water that have all violence in the world. This include wars. And more than 3 million people dying from the air, toxic, pollution in the air. So there is a lot of problems with environment pollution that we need to solve it. And there is a lot of different conventional methodologies that can detect precisely and very sensitively the presence of such kind of toxicants inside the water and air samples, but most of them are very complicated, time-consuming, expensive, cannot be taken from outside the lab, and I think cannot measure, of course, in real-time modes and continuously. But the biggest problem of all of these technologies is cannot predict the biological effect of such kind of chemicals. And it is a problem because chemicals with the different structures can produce a similar biological effect. And in some cases, when you develop a system to detect toxicant A that produces some, I don't know, toxicity, biological toxicity, you can miss another different chemical with the same toxicity effect. So we need something that we can not only detect the presence of the toxicant, but also evaluate the possible toxicity effect on the humans. So there is a huge need for such kind of fast, cheap, real-time and continuous, that can continuously in real-time monitor the presence of the toxicants in water and air. So biosensor can do that because most of them are very sensitive, very cheap, very portable, simple to operate, and continuously it can allow continuous real-time measurements. So what is biosensor? Typical biosensor is built from 3 different sections. The first one is a biorecognition element, interface introducer. So here I'm showing 4 the most common bioreporters: enzyme, microorganism, monomolecules, and DNA. What such elements are doing, they are changing with the presence of the target analyte of stress. They're changing their physical properties. In this way, they're producing signal. So the second section is interfaces, immobilization technologies. We needed to, in order to mobilize the bioreporters above the transducer. In this way, that it will maintain its activity and stability. And the last one is transducers. The transducer, what is they doing? They are measuring the signal from bioreporters and convert it to the measurable signals, to the numbers that we can count. So in this project that I'm going to present today, we're using genetically modified bacteria, wall cells, and optical sensors that are measuring light activity from this bacteria. So what we are using, as I said, is bacteria that was genetically modified. The source of the light is from marine bacteria, and such kind of reporter genes that calls laxaperon is built from 5 different genes. 2 are constructing enzyme luciferase that are producing a light, and 3 is supporting enzymes that producing substrate for the reporter enzyme. And in general, there is, so in this way, if we putting these genes inside bacteria, we can make bacteria to glow. In general, there are two different approaches. The first one is the light off. We're placing genes inside the bacteria that always will produce light. And only with the presence of the target chemical, some toxic pollutants, it will stop light activity due to damaging effect. And if it's not damaging, it will continue to produce light. The main disadvantage of this approach is that it's not specific. We don't know the source of the damaging. So every chemical that are producing light, that is damaging, will stop light activity. So it's less preferable. And what we are doing in my lab also, but what I did before, we use bacteria that modified to sense specifically the stresses. The specificity of the sensing, it's by entering in front before the reporter genes, we're pressing promoter, specific promoter that will activate gene only with the presence of the target analyte. So it means it's called light on mode. In this way, the light will be induced only with the presence of the target analyte of the pollutants that we are looking for. So there is a specificity, as I said, is based on the promoter specificity. And we have many different, we can create many different strains, that each strain depends on the promoters that we are choosing. It's the same promoters that we can find in genome of bacteria. It's gonna be entered in the reporter gene and create, so we can see the list of the different strains that we have in my lab. Each one may be sensitive to some chemical. Maybe it can be sensitive to the stresses like genotoxicity, cytotoxicity, some, I don't know, fatty acid pathways. Okay, so as the first applications that I'm gonna show today is fiber optic based applications for water toxicity monitoring. So fiber optic is built from 3 sections: the core and the cladding with different reflection indexes, and the third one, the black, I'm showing like the black mark black here, is a jacket to prevent the environmental damages on the fiber. So if we will immobilize bacteria above the core, and bacteria will be produce light, the light gonna be penetrate, gonna be go through the fiber optic due to differences in the induction flux. Differences, the fraction induction factors of the cladding and core, and going to be propagate through the fiber to another site. If we're going to place the photodetector in front of the fiber, it will collect the light that comes from another side of the fiber. Okay, so we immobilizing our bacteria using calcium alginate hydrogel. We're just mixing alginate with alginate and dipping inside the calcium solutions. In this way, you can create a hydrogel above the fiber. It can be as a whole fiber coverage, or it can be immobilized with the calcium alginate bits. So what we're gonna do, we're gonna take our fiber, we're gonna immobilize bacteria above it, and we're gonna be placed inside the water. And we're gonna, other side of the fiber, we're gonna be placed in front of photodetector, a PMT camera, and all the measurement process gonna be proceeded inside the black sealed box in order to prevent the light interfaces. We want to collect the light only from the bacteria, not from outside of the box. So how we're gonna use it? So it's very simple. You just need to place your fiber with bacteria inside the water flow. It will continuously monitor the presence of the toxicant inside the water. So with the presence of the toxicant, it's gonna alarm, it's gonna say there's something wrong, and you can close the switch off the water flow and to prevent the entrance of the toxicant to the water stream, like to stop the exposure of the toxicants. So what we did, we just create some flow system, water flowing from one side to another, and the bacteria with the fiber optic are placed inside the water flow, and we just we can control the entrance of the positive controls, some toxicants. We can control the exposure time, concentration to see how it's affecting the bacterial response, if we can see the differences. So at the first stage, when we try to measure the presence of the toxicant, we saw it's not working. We're exposing our system to some toxicant, but it's not decreasing, it's decreasing with the time. There was no light responses. So maybe the reason, because bacteria is still alive, it's need nutrients to make some biological processes. So what we did, we just add 5% medium inside the flow rate and hope we can detect that the bacteria is reacting to the presence of the toxicant. So then we just tried two different chemicals to test if our system can detect it inside the water flow. We expose it to the metamycin C. It's very dangerous mutagen, and p-chlorophenicol. So you can see here the system, our system can detect the presence of this chemical inside the water. It's not only was able to detect it, it also was able to detect it in the dose-dependent responses. The big advantage of our system that bacteria, if bacteria damaging with more damage, it will produce more light according to the damage. So it's not only we will be able to detect the presence of the toxicant inside the water, we will be able to detect its concentrations. So we tested with some environmental water, and we saw that the system, we tested for 24 hours, and we saw the system are able to detect it. We exposed to the positive control twice at the beginning and after 24 hours. So you can see at both exposure time, system is reacted, but the second time the reaction was much lower. The possible reason for that is maybe it was biofouling developed above the hydrogel and prevented diffusion efficiency, and it will reduce the diffusion efficiency of the chemicals from the water inside to hydrogel. And another more, I think, more reasonable like what's happened inside is that a hydrogel is very sensitive to the physical damages, and water flow is just slowly removing the hydrogel from the fiber. So after 24 hours, much less cells left on the system, and therefore there was much lower response. So in general, the system was very fast responding 1 hour after exposure, RO real-time monitoring, and can be used as a yearly warning system in different drinking water systems, distribution systems. So we wanted to solve the problem of such kind of drastic decreasing in the light activity after 24 hours. So we developed new flow system. In this way, there was no direct effect of the water flow above the immobilized bacteria. As you can see, it's protected. It's few holes that allow the water circulation inside the flow box, and so it's much more protective. And indeed, when we tried with the different, first we tested the stability of alginate. We compared the old setup to the new setup, and we saw that the new setup much more stable with increasing in the flow rate. It's the first indication that indeed it's affecting on the, we reduced the physical effect on the membrane. And okay, so we, of course, it's also because, for example, in Europe, you cannot use genetically modified organism outside the laboratories. So in this case, if we're not damaging and not preventing leakage of the bacteria outside the immobilization membranes, it will be able to also to use it in the Europe after it. And then we measure for 72 hours. By in each hour, at beginning of the each hour, which are not 24 hours, 72 hours. And each hour we place some positive controls, different positive controls like copper, ethanol, and p-chlorophenicol. And we saw that system was able, using the new flow unit, was able to monitor it for 72 hours. Indeed, there was decreasing in the light activity, but system still reacted much better compared to the previous setup. And we measure some environmental water like fish, and you can see it also can detect toxicity in the environmental water like fish river and Ashdod. So when you're developing biosensors, you need to, during whole development processes, you need to increase the functionality of the system. You need to optimize it. So I'm here to explain different immobilization, different optimization steps that we did with the system. The first one, to increase, again, the mechanical stability of our immobilized bacteria, we added to the alginate above the fiber optic that we're immobilizing, we added another layer of polysulfone. And it's the main advantage of such kind of additions that it will increase the stability of the alginate from the other side. It also is very corrosive. It's not preventing diffusion rates from the chemicals outside to the inside to the bacteria, but from another side, it prevents the leakage of bacteria from the membrane outside. And we just compare the alginate, unmodified and modified alginate membranes, and we saw that we still, the exposure, the reaction pattern of the bacteria is changed, but we're still able to detect the presence of the toxicants, like the metamycin, as picomolar concentration. It's very sensitive. Another optimization that we did with our system, we wanted to increase the light strength, the capability of our system to produce more light at the same number of bacteria. As more light will be produced, as lower concentration we will be detected in the water. So we're using here metal-enhanced bioluminescence phenomenon. It's based on the physical phenomenon that if you placed the molecules that produce light very close to the surface, modified with nano islands, in this case it can be silver, gold, and in this case it's silver, but it can be also gold and other metal. If it's going to be very close, less than 10 nanometer, the electrons on such a light active molecule, instead to produce, not instead, in parallel of production photons that we can see as a light, some of the electrons going to be passed as plasmons to the surface with nano islands. And we're going to wake the nano islands in this kind of way, is going to produce more light. And we use it to test with bacteria. We place bacteria. We use the same setup, the same black box. We just took we place bacteria on the silver treated glass and untreated glass. And we compare the light activity of both parameters. And we saw when we are placing bacteria above the silver treated glass, we increasing more fivefold. Increasing in the light activity when because it's close enough to pass that the molecules will pass the plasmons and will induce the light activity. When we added silver, when we added another layer of silicon between the silver and the glass, we increased the distance. So the efficiency of the plasmon transfer was much lower. As I can see, it's also a light intensity also decreased. So it's proving this capability. So again, it's very simple modification. By just adding the surfaces with silver nano islands, you can increase 5 times the light activity of your system. And when you're increasing light activity, you're increasing sensitivity of the system. Okay, so the next subject that I'm going to talk about is air toxicity. Again, the same system, photodetector placed inside the black box. You have the fiber optic with immobilized bacteria. The main difference is that bacteria is not dipping inside the liquid, it's just exposed to the air. and all chemicals are that possible toxicants are diffused from the air inside the inside to the membrane, and bacteria are reacting to presence of such chemicals in the air. So what we did, we exposed it to many different chemicals in the air without direct touch. So you can see, first, you can see that it's reacting to the different chemical differently. And also you can see it's dose-dependent response. So it's proving that we can measure the toxicant in the air using this system. And when I expose it to the different chemicals or materials that you can find at your homes, outside, so you can see it's also can measure such kind of presence in the air. And again, in dose-dependent responses, this means more concentration, more light is produced by the bacteria. So the next experiment, what we did, we just placed our black box on one side of the room, and we spilled some chemical on another side. So you can see that after, in the case of chloroform, after half hour of spilling, we already can sense the presence in this chemicals in the air. So this sensor can be used as a air toxicity sensor indoor or outside door environments. It can be again used as a early warning system to monitor the air quality. You need to understand that most of our time was spending inside the building, inside the rooms, more than but nobody understands the real toxicity situation here, because everything producing toxins, everything, pictures, furniture, everything, and when breathing it. So I think such kind of system can say if it's safe to stay inside the offices or home or laboratories. Okay, so using such kind of system, when I'm talking now on the relevant to the post-harvest, what I'm doing with the bacteria, I'm trying to catch, okay, I'm gonna start from the different point. Every fruit or vegetables produce small organic molecules, volatiles, due to metabolitic activity that going from the fruit to the air. So outside the fruit in the air, there is pattern of hundreds different volatiles that producing means stable, normal situation, like pattern. So if some pathogens are starting their activity inside the fruit or vegetables, this pattern has changed. It means the new chemicals are produced, or different chemicals concentration can observe. So if we'll be able to catch this change before on the first step, we will be able to monitor the fruit quality, and we will be able to deal with this problem before a visible effect can be observed above the fruits. So what I did, I took the same calcium alginate membrane, immobilized bacteria, and used for the detector CMOS photodetector, the same photodetector that you find on your cell phones. And I just expose it to the air outside the, I used the oranges. So you can see from these figures that the upper one showing the changes of the light activity of the 4 different strains sensitive to the different stresses to the infection process in untreated, like uninfected and infected origin. So you can see the differences in the positive and the infected items. But if you compare to the real situation, real pattern of the volatiles, you can see that the change is totally correlated to the changes in our setup responses. So we can measure the presence of the infection before we can see it. So yeah. Okay, so I talk about but you can use both cell-based biosensors also on biochip. So if you will take some biochip, you're gonna immobilize above it bacteria, and you place photodetector in biochip, the silicon-based biochip. and you put photo detector, you'll be able to, in small sized device, measure a lot of different parameters. So, so you can use different photo detectors to measure that activity. So you can see when we try it, first we try to see if we see the differences between negative and positive control. Indeed, we saw such kind of differences, and then we try couple different toxicants, and yeah, we can do this. It's not a problem. Okay, and okay, so finally, we can use our bacteria with commercial available devices, plate readers that you can find in every laboratory. It's the main advantage of such kind of systems that it will able to measure 96 samples at the same time. So just need to take your sample, to take bacteria, liquid unmobilized bacteria, mix them together, place inside the 96 well plate, and measure it. In this way, you can measure a lot of samples at the same time, and more. What is more important, to create specific fingerprints according to the differences in the different bacterial responses. For example, if you taking 3 strains, each one sensitive to the different stresses, genotoxicity, cytotoxicity, and quorum sensing, and you expose them to the antibiotics in the water. So you can see every strain responds differently to different antibiotics. So when you are compare them together, you can see you can create specific fingerprints for every antibiotic. In this way, next time when you're gonna measure some unknown sample with bacteria, and they gonna give the same pattern, you will know that it is tetracycline, for example. You can create light fingerprints for each chemical that you are testing to create libraries using the bacteria. If you're using not 3 bacteria, but 60 bacteria, it can give precise determination of the chemical that you're looking for. And couple examples that we did. We test with such kind of system. We test nanoparticles toxicity. We use different metal nanoparticles. We tested some like constructed nanomaterials. And as you can see, that each time when you're using different strains, each time we are getting different pattern, as proving that we can create fingerprint for such kind. And first, using this system, you can not only detect the presence of the toxicant, you can also see the effect of the different modification. For example, size. If you can size, you can see how the size is changing its activity, how size is changing its biological activity, how it's changing toxicity. So you can see the system can detect it. So moreover, you can see effect of the different environmental, how in different environmental toxicants affecting different parameters. In this case, when you're exposing bacteria, let's say only to polymyxin B, it's antibiotic, it's killing bacteria at this concentration. If you're exposing to some metal nanoparticles like copper, but it's not affecting. But when you are adding together, finally you can see that a mixture of antibiotics with metal nanoparticles reducing the efficiency of the antibiotic treatment. This is effect how we can. This is one thing how we can use the system. So why it's important? So if you are taking a antibiotic treatment, and you're living in some area where the pollution, copper pollution, you're breathing it, so it may reduce the efficiency of the antibiotic treatments on you. And for another example, to test the cigarette smoke effect. So what we did here, we bought a lot of cigarettes. It was very difficult to explain to university why I need to buy 200 packages of the cigarette. But we bought cigarettes from the different price range. And we tried to see if the price of the cigarette affecting on its toxicity. using the same system. The system allows to do that. And as you can see, it's very difficult to see, but actually no effect was observed. I think what we are smoking, I'm not smoking, but what people are smoking is the same bad thing, no matter how much you're paying for that. But the main difference is what we saw, the effect of the filter. When in cigarettes, more expensive cigarette, when you comparing the filter efficiency, we saw that the filter efficiency was much more in the expensive than unexpensive, less expensive cigarettes. It means a quality of tobacco is less important than the quality of the filters that you're smoking with. This was the main conclusion from this research. Okay, so we use the same example for cannabis. I'm doing with this Nirit Burstein, this collaboration. And we saw if we be able to see that the composition in the cannabis extracts affecting, we can see the effect of the different composition on its toxicity or its biological effect. So we can see that different extracts with a different composition, cannabinoids composition, affect differently. Again, creating fingerprints that we can use in the future experiment. The system also was able to check it. So system can do everything. It means the toxicity of the different chemicals. For example, buffer toxicity. We used a different buffer. We use the buffer toxicity with a different cytotoxic or genotoxic strains. So you can see almost no effect was observed. This in previous experiment we did with Elena Poverenov, collaboration with Elena Poverenov. Chitosan toxicity. Chitosan, Dr. Poverenov using chitosan as a carrier, as a nano carrier to carry the different fertilizer, biological things to the plants. So we tested it and saw how is the size of the modification it affecting. So you can see the system allow to allow us to understand the biological effect, because after putting to environment of fertilizer, nano carrier with fertilizer, you don't know what's happening with the chitosan, how it's affecting the microbiome around them. So you can see here we can evaluate it. It can be also used in the food industry, not only in the environmental testing. For example, we tested the biological effect of the artificial sweeteners that people are eating, if it's not toxic. So we saw that artificial sweeteners produce some effect on the bacteria. It means when you're eating or drinking artificial sweeteners, you don't know how it's affecting of your microbiome inside the gut. So we test clear, like pure artificial sweeteners, and so there's some effect. As you can see here, with increasing the concentration, you increase or inhibition or induction or inhibition effect of the different, you can see genotoxic effects, cytotoxic effect of the microbiome. And then we try to see the sport additives. We took different sport additives and we exposed to other systems. We saw the similar depend of what kind of artificial sweeteners are used inside the sport additives. Sport additives, sport drink additives. When you're doing sport, you're additive, you're drinking. So depend of what artificial sweetener uses, they're showing similar effect on the bacteria. It means it's also affecting. We don't know how, we don't know, but it's affecting on the human health. So another, I guess few times, but we tested so many things, water quality of the different rivers, lakes, Amazonas, chlorination effect that we're doing with Michael, effect of the processed meat, if processed meat as toxic. It's surprisingly it is. Nanoparticles, plastic, pesticides, nano, like everything. It was twice nanoparticles, antibiotics. So many things. We can use the system to detect newer antibiotics. So many papers was published with many collaborators. So to conclude my talk, this system is very simple to operate, simple to modify. You can create a bacteria. You can create genetically modified bacteria for everything. if you know how it's affecting cell, you can isolate the promoter and use this as a reporter in the future setups. It's one-step procedure. You don't need to treat your sample. You need to clear, extract, or do something. You just need to take bacteria that you grow, expose it to your analyte, and test how it's affecting. It's multitasking. It means scan in one setup, you can measure 1,000 different samples at the same time. It's high throughput. It's showing not only presence of the toxicant, but also biological effect on the cells. And also it's very specific, depends on the promoter that you're using. So thank you for all my collaborators, my supervisors previously, that I did most of the job work on their lab, and my lab members now in my lab. Thank you. Question?
Thanks, Evgeni. I have two questions. First, bacteria are not necessarily stable over time, so how do you ensure it's sensitive?
It depends at what kind of setup you're talking, because if you're talking kinetically measurement, continuously, you saw that with some, like if talking about the water, if you're gonna add a very low amount of the medium, you can measure for a long time, because all bacteria that we are using, it's environmental bacteria that are surviving outside, so it can survive. And we're doing a lot of different things.
Of soil is not environment of water.
Yeah, so you can modify bacteria, any bacteria that you want. You can take bacteria from soil and just modify it to take care capability to produce light. You don't need to use your own bacteria. So it's not a problem.
The other question is how, because you're talking about toxicity, but this is a promoter that's being induced in a bacterium. The question is how strong the correlation is between response that you're getting and cytotoxicity, for example. And for microbiome, if you're saying the microbiome, the gut is mostly anoxic or anaerobic, so the conditions are very, very different.
Okay, first of all, when it can be answered in two ways. The first one, when you're exposing bacteria for your toxicants, bacteria are acting, some gen are upregulated, unregulated. So if you know which gen are regulated by exposure to the chemical specifically, you'll be able to take the promoter for such regulation pathway and just place it in front of the gens. So each time when bacteria will use this chemical, it will react to it, and also it will produce light because in parallel to share the light the enzyme activity of the. Another way, if you don't have enough, if you don't know such kind of systems that is separated gens, you can take 20 different bacterial stresses, each one sensitive to the general stresses, different general stresses. So if you're taking this, they act differently to the same to the different chemical. So for your chemical, bacteria number 1 will be increased 2 times, bacteria number 4 will be decreased 5 times. But together they will create fingerprint that's going to be specific for your analyte, because other analyte will affect differently the same pattern of extreme 20 bacteria. So, yeah, Michael.
Most of our facility going to be exporting research and information to patients much more. And this is from my understanding. Concentration sometimes not so low, like 1 microgram per milliliter. It's like 1 ppm. It's so much concentration, so 1 ppm of water, it's huge. If you have if it's a example, you have all organics like 2 milligrams of carbon dioxide. Talk about 1 microgram per milliliter, it's not very low concentration.
It's a very good question. And I just represented the proof of concept of the system. When you're processing, you usually use a very high concentration in order to see the capability of the system to monitor things. But when this experiment exposed to see the sensitivity of the system. So we saw, for example, for air we saw that it's able to detect 0.8 ppb of chloroform outside text. So it's very low concentration. And also when we optimize the immobilization process, so we pick up a large concentration of the chemicals in the water.
Basically, what you do, you optimize the power. Which means that in lab conditions, it probably will not work. But then we will have mixtures which will be capable of it. You have shown with the antibiotic and compare each one separately, like have no show effect, and let's do like there, the cooperative was not, but together there was strong influence. You might come to explanation, but I just try to figure out why that happened. Yeah. And I was thinking maybe it's because compare forms complex with the antibiotic. Okay, basically stabilized, it does not touch you're reducing killing concentration of bacteria, but you have the effect. So you bring me to the question in reality, you don't know what you have, don't know what you have in the area. Yes. So which means if I have no response, we have response, how do you know what you should do to make it specific for what?
You are teaching system, like every system is supposed to be calibrated in order to functionalize. So you just need to expose to the different composition, different concentration of the chemicals, and you learn, you're teaching the system how, you're learning from the system also how it's responding to such kind of matrix of composition. So you're calibrating it, and then when you are exposed to environmental water, so you correlating to the system library of the exposed reactions that already have been done. Of course, more chemicals you're gonna add, more composition you're gonna add to the library, more efficient your detection efficiency more increased.
One more question, which is probably provocating a bit. I was not very impressed by the situation in chicken water, like water in municipal. Taking that water, very responsive. Took several water samples from different areas, response concentration. There's something that makes a light bacteria which works as a red light system. My question is, basically the approach is now like for a while. I mean, also with the light on effect, it's shown from 2009. So how do you see dynamics in this for 15 years, the last 15 years? It go up, go down, it just don't.
I think like in every biosensor development, there is a critical parameter that's supposed to be done in order to move it to the commercial stages. In general, 10 years ago, there was commercial kit based on the bioluminescent bacteria to test the toxicity, but it was a switch off mode based kit. It means light was all, bacteria was always lighting, and by toxic compounds, the light was decreased. So in our case, couple parameters supposed to be fixed in order to make it commercial, like repeatability of the system, creation libraries of the bacterial responses, creation specific strains for the specific parameters that you're looking for. I don't know how it's only increasing. For now, the interest in such kind of sensors increase with the years. I don't know when it's going to be. When it's going to move to the real commercial like kits or devices, but it's clear dynamic to that.
Where they have a big water supply system, they have a flow cell with fish, on camera on the fish, and tested the activity of the fish, and it goes down to this
camera and microphones also.
Yeah, I don't mind that. So what is the advantage of the
it's very good question also. It's very good question also. Thank you for that. But you need to understand, first of all, when you're talking about fish, you can see only lethal effect of the lethal concentration of the chemicals. In our system, we can see sub-lethal concentration. It means only when it's not came to real dangerous concentration. This is first advantage. Second advantage is specificity. If you don't know when you're using fish, you know that only the problem, but you know what the source of that. You know what effect. If you will know how it's affecting, you will know also how to do it with it. What you're going to do with that. So it's specific specificity and of course concentration. This is the main advantages of the system.
But probably the fish doesn't feel good with the water. It's probably more related to human not to feel good with the water.
Of course, but many studies have been shown very clear correlation between bacteria, prokaryotic toxicity and eukaryotic. Again, a couple like pesticides, for example, that I think some like cannot affect bacteria because they don't have ligands above the so it did. But most of the chemicals are affecting almost similar.
For nanoplastic, there will be different conclusions about the effect on bacteria, including the real effect and there is no any effect. But if you have a fish that is applying different
Exactly.
Questions from the Zoom participant? You can unmute yourselves. Okay. So if no question next week, we will not have a seminar because there is the AR conference. So we will meet again 2 weeks from now.
Okay, thank you very much, guys.